Hi, folks. Welcome to our April 3rd Thursday. I hope you are all on the brink of bud break and that you're all tied and ready. Um, it was a little dicey for us, I can tell you that. Today we have a talk that I know you'll all find really interesting because we are always interested in smoke taint research. This is an untargeted look into how wildfire smoke may change the aroma profile in wines with Carien Catlett, who is a UBC doctoral candidate. She's enrolled as a PhD student at the Okanagan campus of UBC under the supervision of Dr. Wesley Zandberg. She obtained her master's degree in South Africa in the field of chemometric applied to spectroscopic data in the field of fluoro-carbon chemistry. So she's a real thing. She's a chemist. Carien and her family moved to the Okanagan in 2023 from, I'm not going to pronounce it, South Africa. You can tell us, Carien. Thank you. And I'll let you take over.
Thank you, Kathy. It's KwaZulu-Natal is
Yeah, I'm
the
glad I didn't try.
province. Yes, that's sort of on the eastern side of South Africa. Very beautiful area.
Nice.
Thank you, Kathy. I'll take over. I'm not a candidate yet. I am enrolled as a PhD, but hopefully we'll get to the candidacy part later on. Thank you everybody for joining. The title is Untargeted Look, which is what my project is focused on. But in today's talk, I'll also be doing a little bit of an introduction on where the research stands on smoke taint, and a little bit of a very basic analytical chemistry introduction, just to hopefully be helpful that when you hear about research in this field or read some articles, that you understand what that means. Just going to move my face out of the way here that I can see what I wrote. There we go. All right, so the first section is just this short summary. I'm not going to go into a lot of detail. I think everybody has heard many presentations on the history of it, but I just want to recap to get an idea of what we know, what we don't know. And then there is going to be quite a portion of this talk dedicated to some of these scientific concepts. You would have seen these terminology um, a lot, things like chromatography and mass spectrometry. And I'm just going to explain it in a very, very basic way. So for those of you who are in the science field that might have joined, you might find that section a little bit boring, so I apologize ahead of time. And then I want to give you a little bit of a taste of the results I already have from my project, or at least the section of the project. And then I think what everybody's interested in is the last section is just some talk or some summary on the mitigation practices and treatments, what we currently know and what is the advice currently given. Most of that and a lot of the content does come from the Australian Wine Research Institute. If anybody's interested in smoke taint, they have a lot of information on their website. They have been dealing with smoke taint for a very, very long time. And if I jump into that background, we can see here some of the earliest mentions were in 2003 in Australia. And as far back as then, we already knew guaiacol, you would recognize that compound name, and 4-methyl guaiacol as being potential problematic compounds. And so Australia really does have very good resources. Of course, ideally, we want to expand this type of resource to local communities because our bushes look different, our fires look different. But for a start, if you want information, I highly recommend the AWRI. All right, so what we know very far back, we already knew that some of these compounds, like guaiacol, 4-methyl guaiacol, and serindol, are some of the most abundant compounds in wildfire smoke. That's just looking at the smoke, not at the grapes yet. So we know those compounds are probably going to have something to do with our smoke taint. And later on, 2007, this was in fact confirmed, as well as some extra compounds added there, that it is in fact these compounds from the smoke that's causing the smoke taint. Now, intuitively, that feels obvious, but it is something that had to be scientifically proven, and that was then done back in 2007. And from there, we have expanded our knowledge on the types of compounds in smoke and what of those compounds can go into the wines. So many compounds have been identified. We'll see a couple of them, but I'm not going to show you any chemical structures or anything today, just the names of them, and you might be familiar with some of them. All right, and we know smoke contains a lot more than those compounds that we will see today. We looking at what is most abundant, what do we know already from previous research about smoky foods, goes into food items, but there is always scope to learn and research more about smoke compounds. We also know that the volatiles from the smoke don't just stay in their volatile form, but they react on the grape berry skin as well as inside the grapes. They react with sugars and they form what we call glycosylated volatiles inside the grapes. Now, usually because of this, laboratories will include what they call free and bound smoke volatiles in your test. And that is because those bound volatiles, you can't smell them and you can't taste them in your grape juice. So when you assess your grape juice for whether it's going to give you a smoke taint wine or not, you might not be able to assess that properly if a lot of those compounds are bound. And so the laboratories will hydrolyze your samples for you and give you an estimate of all of the compounds in there. So that is why you get both those bound and free numbers in your laboratory results. And there, I say, we also know that the yeast can break down, the same as how the lab hydrolyzes those bound compounds to test for them. Yeast breaks down those compounds as well. And so that's why later in after fermentation, sometimes that smoky aroma comes out, which you didn't get in the grape berry. There's also some research that indicate that some of those compounds can also break down with contact with our saliva. So if some of that ends up in the wine and you taste the wine, your saliva can break down those glycosylated volatile phenols, and suddenly you can smell and taste them, which makes sense if you've tasted smoke taint wines. That is often the case, that once it's in your mouth, you taste it much more than what you could just in the glass. All right, the other thing we know, and this is where my project kind of ties in, is that other than these glycosylated compounds and the actual volatile compounds that are the stinky stuff, the smoke itself also affects how the grape makes compounds. So volatile phenols will include also the stuff that's good, the fruity things, the floral compounds, the stuff that you want in your wine. Having smoke on your grapes will affect how the grapes produce those compounds. In other words, it's not just directly the smoke that influences your aroma profile, but also the secondary metabolism changes inside your grape, and possibly also your yeast. We know that the way the yeast breaks down and ferments the grape must change when there is smoke compounds present as well. All right, so we know it's a complex problem, and that's where I get excited, because I like complex data sets and data analysis. So this gives me a good excuse to do some of that. All right, so here, this is typically what you would have seen if you get lab results back in the past. And I wanted to show this, it's just a portion of a table. It's not the whole table from this article referenced here at the bottom. But you can see if you look at the non-smoke, that's the green strips versus the smoke is the red strips. The ranges, you can see that the smoke, those compounds increase, but the ranges are quite wide and overlapping. So say, for example, if you get a value of 8.0 of guaiacol in your Chardonnay, does that mean it's smoky or does that mean it's not smoky? Because it falls both within the range of what is known to be not smoked as well as smoked. And so this makes it very confusing for winemakers to interpret the lab results. So great laboratory in Australia. I have to give them some credit for what they have done. This is the set of compounds that they analyze for their customers. So you can see they do both the volatile and the bound compounds. So this retinoside, that's a sugar-bound compounds. And what they do is they have a background database for specific types of wines in their region. So obviously we can't use their background data because our wines is going to look different. But if their customers send them in a Shiraz sample, they can give them a graph like this. So the gray bars there represent the background. That is what is a normal amount for this compound after having tested multiple wines that is known to be non-smoked. And then the red dot is the customer's wine. That's where they tested it at. And so the customer can then see if the red dot falls within the gray zones. They are likely not going to get smoke taint. If it falls very far outside the gray zones, they are more likely to have smoke taint. So it's a bit of a better way to interpret your lab results. And so I would very much like to see our local labs have this kind of database. We do have a lot of samples available, and we do have samples that's already been tested for most of these compounds. It's just a matter of collaborating with between the laboratories and getting these standards organized. All right, so into the analytical chemistry, it is very simple. So apologies for any scientists that logged on here. You will hear the words gas chromatography and liquid chromatography a lot when we talk about wine research. And the acronyms for those are GCMS or LCMS. The MS is mass spectrometry. And they are very, very commonly used in analytical chemistry for both qualitative as well as quantitative research. So I thought it might be helpful to give a little introduction of these. So when you hear the word chromatography, what it means is we have a mix of compounds here, all those little shapes. Each represent a compound. So I have a bunch of compounds in my wine sample, for example. I'm sending it through what we call a column, which is a tube type device that has some kind of material inside. And so as my chemicals pass through that tube or column, they are going to interact in different ways because they are different chemicals. They're chemically different. These tubes are often heated, so perhaps like slow temperature and then higher later on. And so because of these changes on the column, your chemicals are going to come out on the other side. I hope you guys can see this little animation correctly. They're going to come out separated. So this is a chemical separation technique. And here at the bottom, these little lines is what we call a chromatograph. So in other words, every peak is a compound that is coming out at a different time. Now I put here in a little block. In real life, we have to consider that biological samples are complex. It's never going to be perfect little peaks, just each representing one compound. There's often more than one thing coming out at the same time. There is sometimes little pieces of your material inside the column coming through, or background noise from the laboratory or from your environment or sample preparation. So it's not as perfect as I showed it here, but that is the idea. And this is what it looks like in real life. And so there at the peak numbered 91, you can see in black there, it almost looks like zigzaggy, a little bit messy. And that is exactly what happens in a biological sample. A bunch of stuff is trying to come out at the same time from the column. Or it could be just a piece coming out over a longer period of time, one type of compound. So it can get a little bit messy sometimes. It's not always perfect. From the chromatograph, so from your liquid chromatography or GC gas chromatography, if you have a nice instrument, it will have a mass spectrometer at the end. And so as your compounds come out of that column, it goes into the mass spectrometer and those compounds get broken up, in a rough way to say it. And the best way I can describe it, there at the bottom you see a bunch of broken plates, and on the left a broken cup. Now if you have a pile of broken porcelain, you can tell if that pile of broken porcelain were cups or if they were plates, because they shatter or break apart in a typical pattern. You're almost always going to get the handle of the cup breaking off in that way as it is shown in that picture. So that when you have a bunch of broken cups, you'll know that is what it is. And similarly, a compound that goes through a mass spectrometer will break up in very specific masses or mass units. And then you get these, what we call a mass spectra, and it effectively acts as a fingerprint for that chemical compound. So to recap, for every compound, we get a line that comes out on the chromatograph. And for each line, there is a matching mass spectra, which is like the fingerprint for that chemical. And then this looks very terrible, but you don't have to worry about all the details. This is what we call the NIST library. So it is a library or a database full of compounds and their spectra, their mass spectra. So on the right there, you can see in the red is our compound, and blue compared to it is the compound from the database that the NIST library chooses for us, because it's a really, really good match. You can see most of those peaks are pretty much looking the same. But on the left, you can see that the NIST library also suggests some other compounds. And that's because when compounds look similar, they might break up in a similar pattern. So you can think of it as if I have a pile of cups broken up, one cup might have a little blue bird on it. And so a pile of cups with little blue birds on them will also have that typical handle that breaks off that we see, but it's also going to have some pieces with blue birds in it. So we're looking for, when we try and distinguish which of those chemicals we're looking at, we're looking for that blue bird, so to speak. And there in the green, the little green circle would be our blue bird. So at the top there, number 1 and 2 are two suggested compounds from our library and their mass spectra. And you can see they both match quite well with our unknown mass spectra from our sample at the bottom, the one with the question mark. But there are differences, like that peak that I circled in green. And also you can see the peak ratios is more like compound 1 than it is like compound 2. That's one of the ways we decide which compound we believe we have. The other way is by context. And so compound number 1, if I look it up in the literature, we already know that it is found in grapes as well as brewer's yeast. So the likelihood is that it is compound 1 and not compound 2. And this is what we call a tentative identification. So it is not a confirmation. There is always a chance that I'm wrong here about that compound, but I have done as much as I can with what I have, and I'm fairly sure that it is that compound. You would need a standard to confirm it, though. All right, so what I love, principal component analysis, that is my bread and butter. I'm going to very, very rough, very short description give to you. You'll often see this used in research. What it is, it's a way to analyze your data, to understand your data when the data becomes more complex. So in this picture that you're seeing on screen right now, you see three dimensions. The one is direction T, which I think in my original example I drew this for was temperature. There's a direction P coming sort of outwards towards you out of the screen, which could be, say, pressure of a system. And then a direction S, which I have no idea what S stood for, but let's say it's concentration. And I have a bunch of data points that create the swarm effect. Now, in 3 dimensions, we can still picture this swarm. We can still make certain conclusions from looking at the data. But what if there is a 4th variable or a 5th variable? Suddenly, we can't see it in our human brains. We're pretty good at going up to 3 dimensions. Beyond that's really difficult. And so using principal component analysis is a great way to bring it to a dimension that we can understand. So the principal component, or the first dimension, is the direction, the vector that is in that direction where there's most of the variance in the data set. And then around that principal component 1, you can see there's some scattering still, and that is going to be our second dimension. That is the second most variation within the data set. So I hope that gives you sort of an intuitive idea of how we look at this data in principal components, and I'll show you some examples of what the plots look like and how to interpret them. Here, one of my colleagues, I inherited some data from him, Harris. We'll see some of his data later on as well. He took a whole bunch of different fuel sources and he burned them and he tested the chemical compounds that we generally look in for smoke taint. And you can see in this graph, it's very difficult to make sense of it. It looks really cool with all the colors, but very difficult to make sense out of exactly what we're seeing here and what it means. He also did the exact same thing for real wildlife smoke, wildfire smoke. He went out into the field, collected the smoke, and tested it. And again, it looks really cool, the colors, but what does that mean? When we run that through a principal component analysis, we often get these charts that has 2 types of information on it. In this case, we have the little numbers, which is the type of sample. And in red, we have the compounds. So where you have a bunch of the numbers clustering or sort of gathering around one of your compounds, we say they are highly correlated. So in this case, we can see 1 to 11 is highly correlated with phenol. Now 1 to 11 in that table, we see that's the dates. So those are the real wildfire smoke samples that he took. And those, in other words, correlate well with phenol. That means the real wildfire smoke has a lot more phenol in it than the other compounds that he tested for. Whereas the smoke from the fuel types, when he did that, had less of the phenol. So in other words, there was a difference between his wildfire samples and his fuel samples. We can also see that number 12, number 13, and 14 is quite close physically in proximity to those dated samples, so the real wildfire samples that I highlighted in blue there. And if we look at what is 12, 13, and 14, we see it's our wet plant material that is typically found in our area. And so, in other words, that represents most of what real wildfire smoke should be like. And in the lab, that is what we want to aim to reproduce then when we're doing small scale or in lab tests. And just further to show this type of image, how you correlate things, you can see 8, 9, 10, and 11, the samples at the top there in the picture, is really close to the compound guaiacol. And then we go and look, what is 8, 9, 10, and 11? Again, that is those last dates. So somehow in the last couple of days where we took samples, that smoke had more guaiacol than some of the other dates, which is interesting. Like what changed there towards the end of August that there would be more guaiacol coming off of the wildfire smoke. At the bottom in the green blob there correlates well with cresol, and I highlighted in the table the green there, that is mostly dry material. And that's interesting. These are very preliminary results, I should mention, like don't take it as absolute, but it is to show you how these types of pictures and these types of analysis can be used to make inferences on the data. And so possibly we should be thinking that if dry material creates more cresol and cresol is a problem, then would removing some dry material in the environment help to reduce the cresol emissions in wildfire smoke. All right, then another terminology in the research spectrum would be untarget versus target research. There isn't a better or worse between these two, it's just different. So my personal preference is towards untargeted. It's more of an exploratory type of research. You cast a wide net and look at different chemicals, as many as your instrument will show you, and you try and look for patterns or trends or anything like that. It's not quantitative, so it is qualitative. And the identifications I make that I show you is all tentative. So that is based on how I showed previously with the mass spectra. I did not use internal standards, so they're not confirmed. All right, which is, a bit of a scientific point there. Targeted research is more often quantitative. A lot of the research in smoke taint is targeted, where we look at those compounds that the labs typically test. And there's a lot of benefit to doing quantitative work if you're looking for very all right. Where did you guys lose me?
Just a bit ago, I think your last word was quantitative. I'm sure you've said that many times,
Yeah.
but yeah, I would just back up like, minute sort of thing.
I believe on
Like
this
if put the presentation back up and maybe I can,
there.
yeah, we're not seeing it yet.
Oh, let me just, because it would have kicked me out here. So let me just reshare this one. All right, let's just make sure I'm sharing the correct one here for you. Is
Yeah,
it
so yeah,
showing?
if just start on that third line, maybe, find something new, look for
All right.
differences.
All
Yeah.
right. Well, I'll give a quick recap on this slide. Thank you, Kathy. Yes, and I'm gonna have to do it all in 15 minutes. I'm gonna do exactly what you mentioned earlier.
We'll ask you questions about
Yes,
the part you missed.
please do. All right, so my personal interest is more to untargeted. Untargeted is exploratory in nature. It's like casting a very wide net. We look for a method. Tara at Friend got us a method which gave us lots of peaks. I'll show you in a minute. Um, and I'm looking for interesting trends, whether or not it's telling us something new that we don't know yet. Um, so that piques my interest. That is fun to do. It's not necessarily better or worse than targeted. Each method has their own benefits. Targeted research, you're looking at specific compounds. The majority of the work in research on smoke taint currently is targeted in nature. So they are looking for those volatile compounds or the glycosided volatile compounds inside the grapes or skins. And I can always expand to targeted analysis if I find some new compounds that I'm interested in, and then I want to go and quantify or confirm those compounds. So they are complementary types of research, but because untargeted is exploratory in nature, you're not going to necessarily see in my data sets things like guaiacol and cresol because I didn't go and specifically look for those compounds. I'm just looking at the chemical composition of my samples uh, in a broader way. All right, so to get into my project specifically, I was lucky enough to inherit some samples from my group, from my research group, from the Zandberg lab. Yeva Zig, she worked with O'Rourke's family estate vineyard, and she went out there and smoked some Riesling and Gewürztraminer grapes in 2021. And so she collected those grapes, and then Harrison White, another colleague of mine who used to be part of the Zandberg lab, he made wines from those grapes. And then recently, Tara ran those grape samples through their GCMS instrument, and she gave me that data set, and that's where my part of the work comes in. So a little bit spoiled in that a lot of work has been done for me and the samples were available. And this sample set I can use now as a proof of concept data set to see if my ideas, my methods are going to work, and then build on from that. So I'm just going to today show you the results for the wine, the Gewürztraminer wine samples, partly because the time won't allow me to show you everything, and partly because I would still like to publish on this, and I am still working on it as well. But there were two types of wine, and Eva smoked those bundles of grapes with smoked tents in the field. And then she also took samples from those same rows as control samples. And each wine was, or each variety was taken at three different time points. The grapes just after she smoked them, at harvest time, and then after Harris made those wines, the samples were taken as well. So, in other words, three different time spots for two different varieties, both smoke and controlled. All right, so from where the samples were collected, they were sent to Friend, and Tara ran them on her Thermo Fisher GCMS. It's a very nice instrument, and you can see that chromatograph. It's got lots of little lines, which means there's lots of information there to work with, lots of data to work with. And luckily, my instrument at the Zandberg lab and her instrument at Friend uses the same software, Chromeleon, so she could send me those files. And then I had to use software MS Dial. If anybody's interested, they can look it up to deconvolute and align those. I'm not going to go into the technical detail of these data analysis techniques or processing techniques. At the end of the day, I get an Excel file. And what I do want to show you in this little table at the top, you'll see I have a column called ID, which goes 0, 1, 2, and I skipped 3 because that was a background peak. And then the retention time, you'll see that first two retention times are very similar, but then the mass next to them in the next column, the 55, and now I have to remove my screen sharing banner here. Anyway, those two first top rows in the quant channel, you'll see these different masses. So it means there were two masses or two handles, cup handles, if you will, coming out at the same time at that 6.1 minutes. And that's what they call deconvolution. And so when I later on talk about features, it doesn't necessarily mean a feature is just corresponding to one peak on the chromatogram. It could be that there are more than one feature in one of those peaks in the chromatogram. All right, I hope that's not too confusing. To do a principal component analysis, you have to transform and center your data. And so I did some transformations there that I won't bore you too much with. And then I used RStudio to do the further analysis and to be able to visualize the data set and understand what's going on there. All right, so there is the chromatogram as received from Terra, from Frint. And you can see there's one sample that is my control sample, number 3, the third control sample of my Gewürztraminer wine. And so hopefully if I get Terra some chocolates, maybe we can get a rerun on that sample because it is definitely an outlier. You can see the purple lines there, very, very different from the other wines, which is a little bit unexpected because it should be more similar. But it does give us a chance to see how some of these visualizations help us see differences like that outlier sample. And so in this heat map, the red bar at the top shows us those are smoked samples, and the blue bar at the top is grouping together the control samples. So for the most part, in other words, our data set is distinguishing between smoke and non-smoke samples, which is very good news. And again, just that third line here, that is our third control sample of the wine, that's that outlier. You can see it is quite different. It's a good visual way to inspect your data. Let me just keep an eye on the time there. We're still okay. All right, and so this is a score plot after running the principal component analysis. And here at the bottom, you can see dimension 1 explains 61% of the variance in my data. And this makes me fairly happy because along the first, in other words, the x-axis, along that first dimension, the left side are all smoked samples and the right side are all control samples, which tells me that 61% of the variance in that data set is telling me there's a difference between smoke and non-smoke samples, which means that some of the compounds in that data set has been changed due to smoke exposure, which can possibly give us more of an insight of how the grapes are reacting, how the yeast are changing the aroma profile. So that's exciting to see. In our dimension 2, there's a 23.5, sorry, the second language thing. 23.5% of our variance is explained in dimension 2, which is our up and down or our y-axis. And we can see from where that third sample is sitting there up in the upper right-hand corner, indicated by GWC3, that probably it is because of that very different sample in our set that is now kind of dominating our second dimension in our principal component space. So not ideal to have that. It would be nice if we could rerun that and see if that was some kind of error or if that is actually what is in our sample. All right, this is the loading plot that goes together with the score plot that you just saw. In other words, where you saw all the smoke samples gathering on the left-hand side, these compounds, each of these V with a number is a feature. All right, that's that list that says each feature. So each feature is effectively a chemical of some sort that is shown in our data set. And these that are in red and yellow are contributing more to that variance in on that vector. So we can say that these compounds are correlating well with our smoked samples, where the opposite is true here in the right bottom corner. Those compounds are correlating well with our non-smoked samples or our control samples. And then upper right-hand corner again, that's going to correlate with that outlier sample. All right, so interesting here is you can see both on the left and the bottom right that phenyl ethyl alcohol. So why would the same compound come out in both smoke and non-smoke samples as being important? So there's a few reasons for it. One possibility is that one of those is not identified correctly, that it is not actually phenyl ethyl alcohol. And that is where my job continues from here, is to go into features like that and make sure exactly why is it like that? Is it that compound that NIST originally identified? And this is a list. So this is the most contributing compounds that I identified in the Gewürztraminer wine. That is explaining the variance that we saw. That is distinguishing between smoke and non-smoke. And now two things to note here is one, there's no glycol, there's no cresol, so we're not looking at actual smoke compounds. We're looking at metabolites. So we're looking at how the smoke changed how the yeast or the grape metabolizes the wine must. All right, so most of them you can see they are known to be yeast metabolites, and a couple of them are known to be also grape metabolites. And one or two of them is uncertain. There's one that's, that 1,3,5 trimethylbenzene, which is sweet aromatic, but it's a coal tar component. So it could either be that that chemical was incorrectly identified. It could be that it is contamination during sample preparation, or it could be something that's very interesting because coal tar, maybe that is telling us that that is another compound we want to look at. The flavor that I have in that second column, that is all the FEMA list for those compounds. So you can see there's compounds that you would consider positive flavors, fruits, honeys, that came out as being important in considering the difference between smoke and non-smoke. So we're not just looking at the nasty stuff, we're also looking at how the good stuff is changed due to smoke exposure. All right, so I still have to refine the results and go deeper and try and make sure that the compounds are tentatively identified as best as possible and look into why they might be important. And then we might want to consider confirming some of those compounds by doing targeted analysis after this. And then I want to design a phase 2 where we're going to do more samples, possibly focusing on wine and not necessarily the juices, but trying to get more samples so that we can do a better analysis. All right, so the interesting part that everybody's been waiting for, I believe, and I have a few minutes left on that. I don't need too much minutes on that, is the mitigating and treatment plans or practices that is currently considered best practice. All right, so I kind of listed them here, what I see as sort of back to or front to back mitigation practices. And of course, number one there is fire management. And I am not a expert in that field. I am a chemist, but it makes sense that if we try and manage the fires, then there's no problem. Where there's no fire, there's no smoke, right? And so that can be a discussion, I think, on its own, of whose responsibility is that to manage the risk. We saw there that possibly removing dry material fuel sources could change the composition of the smoke. So it is not a small step. It is possibly the most important one. All right, then after you did have the fire, you did have the exposure, you want to do a risk assessment. Now, generally speaking, it seems that the closer you are to the source of the fire, the higher your risk is going to be. We know that during variation, the grape is most susceptible to smoke taint. So if you have early in the season smoke, it's not that much of an issue. If you have it during variation, then you're probably going to get some problems. The duration of exposure. Let me just move my face out of the way because it's right over my writing. There we go. And then the advice generally from the Australian Wine Research Institute is to use laboratory tests. They're only useful if you can interpret the results. So hopefully we can give better guidance to industry on how to interpret those results. And again, as we build collaboration and connections, we are here as researchers to help industry interpret those results, and of course, talk to your laboratory person who analyzed the results. And then a big thing you will see towards the bottom, I mentioned it again, is the small batch ferment. A really good idea is to do a small batch ferment and see if some of the smoke aromas is coming more clearly out of the wine than it was in the grape because of that glycosylation of the volatile phenols. Now, one section where there isn't a lot of work done on is prevention in the field or at harvest times in the form of barriers, films, covers, washing, things like that. The bit of work that we've done in our lab, we haven't published on it as yet. We have done some work on washing studies, and to be honest, it is not looking very promising, just basic washing studies. So one would have to look more into exactly what to wash the grapes with if you're going to try and wash off some of the smoke taint. There is some a little bit of studies done, sorry, about using a spray film over the grape. But unfortunately, those films have actually worsened the problem. But most of them were oil-based. So moving away from oil-based, maybe looking at a different type of film that doesn't help the compounds go in, but stops them from going into the skin, into the berry is an option. My feeling is that a lot of the research has stayed away from this because a lot of the feasible options for this would end up being very expensive and impractical for the farmers to implement. All right, then harvest protocols. Again, on the AWRI website, they have more detail on that. All the harvest protocols is around trying to handle those grapes gently, keeping them cool, keeping foliage and sticks out of the mix, and minimizing skin contact. So also minimizing the amount of pressing, rather using the free run juice for your wine, because it would appear from research currently that a decent amount of those volatiles sit inside the skin, and so if we can keep the skin separate, that is better for our wines. It's not 100%. There is some of the volatiles that get through into the flesh and into the juice, but that does seem to make quite a difference. Then fermentation parameters. There's been quite a few research articles on that. It hasn't looked very promising. It's very varying based on the variety, your wine, other wine parameters. There's nothing that stood out as extremely effective. But again, using small batch ferments, testing some different techniques on those small batches can give you a good idea before you go and make your entire batch. Varying some yeast. Again, this is where collaboration and communication is going to be super important. The idea of having this label of Okanagan wines or BC wines having smoke taint is a concern for everybody. And so as we test things, I would encourage the winemakers to share that information if they find yeasts, for example, that work well, or fermentation parameters that work really well. So one of the things is to use oak chips, the raw oak chips, not smoked oak chips, and to add some tannins. And I'm going to quickly run through the last 2 seconds or 2 slides here. Um, in terms of treatments, all of those treatments listed there, carbon treatments, reverse osmosis, they also affect the good parts of your wine, the good phenols, the good volatiles, all right? Carbon has been shown to be the most effective, but you have to do it very carefully because otherwise you end up getting a very watery, non-wine-like wine, which isn't very tasteful. There's one study where they used a spinning cone column distillation with carbon, and that showed quite promising results. So in other words, they distilled off all the nice, really good, flavorful volatiles and stopped before some of the nasty smoke stuff came in. Then they treated with carbon your wine that's left over, that's now smoky and gross, with carbon, and then they added back those nice volatiles, and that seemed to have been quite effective. So that's quite exciting. Possibility as a treatment. And then storage, aging and storage aggravates the smoke taint. So the advice is to get the wine out into boxes, into packaging on the shelves as soon as possible. And then marketing again, it's a problem for everybody in the area. Once it's the area is labeled as a smoke taint problematic area, it's everybody's problem. So we have to work together, I think, on this issue. All right, just to show you that carbon treatments, this one is not the one with the distillation, but Dr. Julie Colbert has a video specifically on her research on carbon treatments. And she used various different types of carbons, which is important. It's not all carbon is not the same. And there is a recommendation of dose to stick under that 1 gram per liter, but actually closer to 500 milligram per liter. Otherwise, you get that really watered down wines. And then this is another type of chart. You're going to see the spider charts. And it just shows you in this chart, if you look at the black line there, close to the smoke or Band-Aid flavor, that's the control. And as you add as you add carbon, you are losing that. And in fact, if you add carbon at very low amounts, it seems that some of your good flavor, like here they tested strawberry, is increasing. So the perception of some of your fruity compounds is actually increasing. Obviously, the amount of that compound's not increasing, but the perception is increasing as your smoky Band-Aid compound is reducing. So it's about getting that right dosage of the carbon correct to treat your wine. And so that's where that small batch ferments is really helpful. If you can make a couple of small batches and test different levels of carbon to decide which level is going to be best for you. All right, thank you. And I'd like to give a special thanks to the Canadian Grapevine Certification Network for funding. My supervisor and my colleagues, so Dr. Wesley Zandberg, Yavazek, Harrison White, O'Rourke's Family Estate for allowing us to go and smoke in their vineyard. That's not a small favor that they're doing, as well as Tara Olinsky and Front Vineyards, and Kate and Rue for giving me this opportunity. That's the end of my story.
Thank you, Carien. That was awesome. Do we have any questions? While I wait to see, I think what was super interesting was the fact that the smoke actually impacts metabolites. Right? So that even if you don't have any smoke taint that's perceptible, you have a change there, right? And it's not, and it's because we always think of smoke taint as sort of overlaying, the flavor and aroma profile, right? But yeah, it's kind of a bigger problem than that.
Yes, exactly. It is more complex than that. And it's also not even just the compounds themselves from the smoke. The smoke we saw, and I arrived here in 2023, and that year Kelowna had all that terrible fires. At that stage, I was in Penticton. And so farmers in Penticton might not have worried about the smoke compounds directly getting into their grapes, but there was 2 weeks where those plants didn't get UV. They were really, really hot in that environment,
Right.
and that is going to change your aroma
Mhm.
profile as well.
And also more disease susceptibility, right?
Yes.
Which I thought was just from smoke cover, but apparently UV light inhibits powdery mildew. And so when you're blocking the UV light, you've got more of an issue there, right? We have Lindsay has a question. Go ahead, Lindsay.
Yeah, hi, thank you for that. It's a very interesting talk. Just listening to that, I had a question which is maybe not your area of expertise, but I was wondering, I know and I've had experience with wine that has taint that it does not show it until later, and as it ages, it releases the taint. Has anyone been looking into a way to stop that from happening, to keep those compounds bound to the sugar?
Yes and no. What they looked at was enzymes, in the hope to break it down or stop it somehow. Unfortunately, the enzymes have the opposite effect. It breaks down, and then you have more of those volatiles, smoke volatiles, and there's no way to really remove that once it's broken down. There hasn't been specifically research that I found that keeps it bound, which would be ideal if you could add a food safe additive of some sort to keep it in that bound phase. But yeah, that's definitely also a field where we don't quite understand the chemistry, what's happening inside during the aging process.
Okay, thanks.
Leandro, do you have a question?
Uh, yeah, sorry, I had to jump out of because I have a call, so maybe you talk about it. Have you found any material or you have an experience with the MIPS, those resin products that kind of absorb polyphenols? I know there's a company in the valley that is offering service to treat smoked ones with it. We have tried at the winery, and I had to say we have seen some good results, but I would like to know if you have any opinion about that technology.
Yeah, so I think what you're referring to is what I had to rush a bit through at the end. So one of the treatments would be carbon, activated carbon, usually in powder, and then you get bentonite
No,
spray.
no,
Um,
it is not. So it's I don't know. Maybe you, Kathy, you know what I'm talking about. So Sarah Dweller has a that representing this company. I think it's from New Zealand, and it's a resin or resin. I don't know. Sorry, in my English. So basically, you make
A
all the wine
a resin,
resins.
yeah.
All right, thank you. And this polymer absorb polyphenols, including some of the smoke molecules. As an advantage versus RO is that you don't have to put pressure on your wine. It's just like a low pressure just to make it go through the machine. So yeah, I was curious if you have any experience with that.
yeah, I didn't read specifically on that resin. I did come across an article that they also use polymers, and then they try to label them with a compound to target the smoke volatiles as opposed to the positive volatiles. Um, it wasn't as effective in its targeting ability, so it did take away some of the fruity, floral, good aromas. But again, similar with the carbon treatments, it does seem to help a lot. If you lower your smoky volatiles just a little bit, suddenly the perception of your good compounds increases. So yes, they do. There are quite a few products that does seem to be effective.
Thank you.
Thanks, Leandro. Carien, thanks again. It was super interesting and a lot of science. A lot of math too, although I know you didn't bring that up, but
Yeah.
yeah, I know all that calculus came in handy for that. Um, So if you have further questions for Carien, you can Kate will send off her contact info, and you can contact her directly. This video will be up on our website for your friends and acquaintances to see. And yeah, thanks again. So Kate is going to send out some links to some surveys that the government is conducting, mostly on reaction, I guess, to climate change and, etc. I know we're surveyed out, but these surveys are really important because they help determine how much funding the government gives to assistance as well as research in terms of climate change and climate resilience. So please watch for that and tell your friends, et cetera, et cetera. Carien, I think we're going to have you back. So good luck with your further research. And
it was lovely to be here. And I think if there, if from industry side, if there are specific topics that they want us from research to clarify, we definitely always keen to have those conversations. There is a conference, a student-led conference at the end of this month on the 30th of April with the Wine Research Council. And so I'll be there as well. And then I'd also like to give a shout out to Nisa Chevet. I'm sorry if I'm butchering your surname there, Nisa. She is with BC Agricultural Climate Action Research Network, and so she is working on a research brief specifically on mitigation and treatment. So everybody can look out for that being published probably towards the end of the year, Nisa. I'm not sure if you're still online. You can let us know.
Awesome. Thank you. Thanks, everyone. We'll see you next month on the third Thursday of May, whatever that date is. And Kate will be sending that information out to you. Okay, bye-bye.